hopefully.
Yep, looks good, Matt.
Okay, so I've got just a few minutes to talk a little bit about the Helicase project and how we're doing the open bit and how we're trying to use that to build a community of people to solve problems. Just to go over a few of the really basic rules that underpin everything. We've been doing open science, I guess, for a while now, and the basic idea is that everything's shared and anyone can take part, and there's no patents taken. And this has been used for a number of studies over the years, and this kind of level of openness underpins a lot of the stuff, of course, that the SGC does. Over the years, we've identified that the things you need for that are, lab notebooks that are open and online, and places where people can interact, good places where you can put open data, and social media for building a community. So they're pretty well-worn components now. And I guess anyone who's worked in open science and done it this way knows that if you are open in this way, that you I'm preaching to the choir here, aren't I? If you are open in this way, then you tend to get really high quality inputs to projects, provided people know that their contributions are going to be valued and they're needed, so that people can really take part as key participants. I think it's very important to deal with the slightly dull thing of licenses, about making sure that everyone who takes part knows what the freedoms are that they have. And open science means so many different things to so many people. My colleagues think that open science just means we're publishing open access papers, and it does help to be a little bit more specific about what you can do. And I think one of the advantages of open projects is that people don't have to keep on asking for permission for things. They know by virtue of the license that they're allowed to do certain things. Of course, it's one of the powerful things about Wikipedia is that it has a Creative Commons license, so it makes it very clear what you're allowed to do. And all the stuff that we do in the MedChem project, MedChem core of the READDI AViDD is governed by a Creative Commons license. So you can do various obvious things with it, and you know where you stand right from the get-go. The model in terms of developing open assets and then thinking about translation is quite clear, I think. There are lots of ways of thinking about how you might translate open things, but in this project, the idea is that we get to essentially a probe molecule with a lead CV with a bunch of criteria hit, and then we stop in the MedChem core. And that opens up the possibility that things that we develop, probes, can be translated by our commercial partners, our private sector partners. So the idea is that a probe still has enough chemical space around it to allow for something to be commercially developed, if that's the route you want to take. And there are good examples of that from the SGC over the years of how chemical probes have led to the development of commercially viable molecules. In terms of thinking about these open chemistry networks, which are how we're organizing the more open and community-facing stuff that we're doing, I guess that over the years we've built up a fairly good understanding of the components that are needed for that. From the way that we meet online and live, through the need for raw data to underpin everything, the need for champions of various kinds, which I'll talk about in a minute, and the need for publicity and outreach to make sure people are aware of what we're doing. One of the core technologies that can help with that, with sort of people understanding what you're doing, is the use of very kind of radically open technologies, such as open lab notebooks that are machine readable. So one of the things that we found is really quite powerful are lab notebooks like Lab Archives, for example, which is one of the ones that's available and which we use for a bunch of projects, where all the entries in the lab notebook are Googleable. There aren't many lab notebooks that allow that, but Lab Archives is one example. And what that means is you can search using Google for specific molecules or UniProt IDs or whatever you want to do, and use that to discover the science. Now that's important for community building, but I think it's also important for machine readability of what you're doing. So if you're working on molecules, working on a chemical reaction, it can be found by an AI pretty simply. And that can really help grow the project and connect you to others, I think. So specifically for the Nsp13 project, we use a website called GitHub for a lot of the discussions, which is a powerful big website that hardly any scientists use, but a lot of computer scientists use it. I'm sure it'll be very familiar to a lot of people on this webinar. We have a pharmaceutical industry champion who attends meetings and gives strategic advice on this project. That's James Day from Aztecs. we're a fragment-based project, so it makes sense to have someone like that on board. A student champion who deals with lots of things to do with meeting organization and making sure people are pursuing their action items and stuff like that. Lab notebooks, as I mentioned, social media for community building. In this case, the SGC's Twitter feed is pretty good for communicating what we're doing. Each month, the meetings are recorded and put on YouTube. And as a result, we're already seeing collaborators coming in, giving synthesis suggestions, giving help on assay development and things like that. So as was briefly mentioned, we are using as a starting point this wonderful paper from Joe, from Frank, which details lots of fragments bound to the Nsp13 structure. And in that, there were 3 groups of fragments that we thought were quite interesting. 2, I think you've heard about already, but there was a site that we called Site 3 up on the periphery of the protein, where there was a little cluster of 5 fragments that were bound. And based on what Tim had just said, we found this idea quite interesting, that we would target a sort of peripheral area of the protein in case that was that could potentially lead to a spanner being thrown in the works of the motion of the protein. And so we started by simply ordering molecules and designing molecules. Did a little bit of fragment soaking on some of these things, so we built up a pharmacophore model, and then tried to grow and merge some fragments. Didn't pursue that too hard. We got some molecules tested by Joe at Diamond, and no hits from a really simple way of just sort of merging fragments. But use that to do a more complicated analysis, I guess, of using the pharmacophore to define the pocket of interest and then essentially ignore the fragments and start from scratch. In this case, doing a docking of the entire enamine real space library, as you might expect, but also trying to use a more generative approach in addition. So training an AI on something like Chembl to spot molecules that are potentially bioactive, and then sort of doing an iterative generative approach guided by the pharmacophore to try and design molecules that should bind. And I guess so we're interested in testing out which approach is better. Do you just go for the commercial set based on a docking experiment, or do you go for a more generative approach with a trained AI where you start from scratch and use the training to get over the problem of an almost infinite amount of chemical space? And I guess that's what we're doing right now. So we're deriving commercial and in-house synthesized molecules for both of those classes and seeing which one wins. So these are the current ones that have been bought from enamine, and we're supplementing the generative set, which is a little bit more difficult, with some commercial synthesis from CROs and in-house synthesis from various people, including my PhD student, Tom. And we'll see, which is the better approach. To assess the molecules, we've been trying to develop an SPR assay, which has not been that simple. So protein was developed by SGC Toronto that was biotinylated, and we're using strep chips. And this data is from just last week, where we've been trying to show that the SPR assay is functional. Made a little bit difficult by the fact that, as Tim mentioned, there's no really good positive control. There is this complicated looking compound here that was published quite recently in the last month or so, I think, which comes from pomegranates and is an incredibly tight binder of apparently this protein and a bunch of other things. And at the moment, we're using that as a kind of control, even though we're not quite sure about its specificity for binding. But the last trace that we got just last week does indicate that we're seeing the right kind of binding. So we think that the SPR assay is working, which is good. And a preliminary evaluation of some of the molecules that we have bought, as I mentioned, from the docking of the enamine library, as well as 2 molecules on the top left there that came from a targeted library we synthesized from one of the papers that Tim mentioned, has thrown up some interesting and preliminary data, which we're quite excited about, that these molecules do seem to be hitting on the SPR assay against the protein from SARS as well as from MERS, and show a little bit of a sniff of activity in an ATPase assay, which is also up and running. So early days, but it's nice to have early breaking structures to show people. And as I mentioned, the SPR assay seems to be working pretty nicely. So Tom in my labs are working on it, and Samira in Toronto has been pioneering this, and it's going to be a useful assay for this project as well as we think obviously for the CASH project. So essentially, we are live and online, and obviously everyone's invited to participate. The license is there. We are trying to spread the word. This is a picture of my lab in UCL, and usually it's a ground floor lab, so you can kind of if you stand on that wall there and look in, you can see the molecules we're making. And we put little posters in the window to try to encourage people to participate. So as you're on the street, you can see that QR code with your mobile phone and get taken to the relevant page. You never know who's walking by the window. About to hire a bunch of people to help with the AViD project, which is good at UCL. We always need volunteer pharma champions to help drive some of the community projects, about a 3 hours per month time commitment on average. And if you'd like to come along and see how it works, the next meeting is going to be on December 12th. And there's a link there for you to see all the meeting recordings and minutes and stuff like that. So thanks for the chance to come and talk today. Thanks to these funders and some more people for their many and various inputs. And yeah, thanks for your attention.
Thank you very much, the READDI team. Thanks so much for this very exciting talk on Nsp13. Really exciting target, and really appreciate and admire your effort in bringing open science alive for Nsp13.